Yes.
Okay, perfect. So today I'm going to talk about the work that we did and it's recently published in Genome Research, where we use deep learning approaches to get more insight about RNA binding protein target preferences. So I'm pretty sure that you heard about deep learning because now it's everywhere. It has transformed fields such as computer vision, speech recognition, and natural language processing. Now it's becoming the method of choice for many applications in genomics as well. And to explain the advantage of deep learning compared to traditional machine learning, let's say that we want to create a method to distinguish the images of the dog and cat. Can you see my cursor? Yes, okay. Given a set of image labels of cat and dogs. So in traditional machine learning algorithm, one needs to first extract the relevant features manually from these images, like the eyes, the nose, the ear, and so on. And then the classification model uses these extracted features to classify the image as a dog or a cat. And this is not the case just for the images, and this is the case for all other kinds of input like speech, text, or genomics. And for example, in genomics, one common feature that is used for machine learning algorithm is K-meres counts from the sequence of the introns. So because of that, the classification performance of these models depends heavily on the quality and the relevance of the extracted features, and the incomplete representation of the data may reduce this classification accuracy. And deep neural network or deep learning solves this issue by embedding the computation of the feature into the model itself. So it takes raw data as input, and it can learn all the features from the raw data. However, to train these neural networks, we need lots of data. And actually, the training of deep neural networks have been enabled by the huge amount of data, in addition to the computing power and, of course, algorithmic advances. So what is actually neural networks? So the basic building block of neural networks is an artificial neuron that it takes as similar to biological neuron. It receives some inputs and performs simple but nonlinear computation and produces an output. However, the key is that when you combine many of these neurons in multiple layers and feed data through this multiple nonlinear transformation, then each layer learns a different level of abstraction, and at the end, the model uses the high level features to classify, for example, the image to a dog or a cat. And a popular class of neural network are a convolutional neural network that introduce a huge breakthrough in image recognition. And the major building blocks of convolutional neural networks are convolutional layers that we can consider them as some filters that scan the input to detect a specific type of the factor. For example, in the face recognition task, an image comes in the forms of an array of the pixel value, and the features in the first layer typically represent the presence or absence of the edges at particular orientation, and then the second layer starts to combine them to represent features that correspond, for example, to the parts of the face, like nose, eye, and so on. And the subsequent layer would detect objects like faces as combination of these parts. And the key aspect is that these methods or deep learning approaches learn these features through the training. So it's not a human engineered features. And this convolutional neural networks are also widely used in genomics now, for example, to find the binding site of transcription factors or RNA-binding proteins. Usually the input is the DNA or RNA sequence that is encoded to a matrix of 0 and 1, and the neurons in the first layer can be considered as a motif finder that scan the sequence and score the input. And the next layer, next convolution layer, starts to combine these motifs together. And at the end, the output, the model uses these high-level features to distinguish between binding site of between the positive samples or negative samples of one RBP. But this the output is not necessarily just one output, so we can learn the binding site of different different proteins at the same time. And the other nice features of deep learning approaches is that you can also integrate multiple data into the model. So you don't need to just have one input. You can also learn from multiple output, multiple input data. So as I said, because these methods learn high-level features abstraction, they have a very good performance. But for a long time, they were considered black boxes because it was not clear how they make decisions. And in many applications, the label is the most important thing, but this is not true when we are making, for example, medical diagnosis, or when we want to extract new insight from like complex data such as genome. So when the model interpretability is important, simple methods like linear models were more desirable. But now scientists are developing methods to open the box and see what's going on in the box and what we learned from the data. And there are different approaches, and one class of these approaches focused on feature attribution methods. They compute which features or what features in the input are important for a neural network when making a prediction or on a particular data point. For example, or they can also like answer the question like how much each part of the input contributes to prediction. And by doing so, they can assign a score to each feature in the input, like indicating how much this feature was important for the prediction of the output. For example, here in this case, you can see on the right figure that the part of the image that were important for the model to classify this image as a camera. And so we use this kind of deep learning approaches to get more insight about RNA-binding proteins. I'm pretty sure that you know that RNA-binding proteins play important roles in all aspects of post-transcriptional gene regulation, from splicing to translations. And we have now technology to assay the interaction of regulatory factors on RNA, as we do also in DNA. Called CLIP-seq protocols, or cross-linking and immunoprecipitation followed by sequencing. So there are different CLIP protocols, and I'm not going into the details, but very, very briefly, they first cross-link the RBP of interest with the protein, and then the protein RNA complex is extracted by an antibody that is specific for the protein, and then the extracted RNA are being used to input for the sequencing facility. And then we got like this rich fragments, and we can map them back into the genome. And then by using some peak calling algorithms, we can find the peaks or the potential binding site for each RBP. And we use these peaks or binding site obtained from these peak calling algorithms for our study. So More specifically, we used Parklip data sets or the peaks from the Parklip data set from 60 RBPs in hexal line, as well as peaks from eCLIP data sets generated by ENCODE for around 150 RBPs in 2 cell lines. And so just to make sure that the data sets were processed with the same pipeline for each data sets, and yeah, everything is same. And then to define the input for the model, we first bin the genome, and each bin is considered as a binding site of or positive sample of one RBP if it overlaps with its peaks, and otherwise it's a negative sample. So for each bin, we have a vector where is the vector of 0 and 1, when 1 means that this RBP is a binding, this bin is a binding site for the RBP, and 0 otherwise. And for the input of the model, we use the two different information, the sequence information, like just the sequence around this bin, and also the annotation of the nucleotides around this bin. So as you can see, like if these nucleotides are in the CDS or the coding part, intron, 3' UTR, and 5' UTR.
So Mahsa,
yes.
Were these the only raw input data that you tried, or were there others and you zoned in on these specifically because they were informative? And if you could speak a bit more about that.
I'm not sure if I really understood your question. So these are just like, these are the peaks that comes from different Parklip data. So from Parklip or eCLIP data. And so these are the peaks, and then we bin the genome, or like better say like transcripts, and then if this peak overlaps with this bin, then we say that this bin is a binding site of the corresponding RBP.
Yes. So what I was asking is, you are looking at sequences and region types, but there could be other data sources like conservation that could be informative for binding peaks.
Yeah. So we are actually working now on adding the structure information because we know that there are some RBPs that like to bind to the RNA, like the structure motif, but yeah, we haven't published that yet, but it's not a really trivial task. But yeah, for sure you can add as many data sets that you want, because this is one of the characteristic of the deep learning, that you don't really need to have just one input, and you can just like integrate a different kind of input.
Okay, so we have an audience question as well that is related. So Irene asks, for the ENCODE eCLIP, there is a lot of binding on non-pre-mRNA elements. Did you focus only on the exon, intron, and UTRs?
Yes, yeah. This is also a part that we focus on the exon, intron, and the transcript. So mostly like pre-mRNA.
And there is a specific reason for that, or reasons for that?
Because it's hard to like distinguish whether it's like binds to actually the mapping is like transcriptome wide, so we didn't really map the RNA fragments to the transcript. We mapped them to the genome, so it's not like trivial to just like do this also.
Okay. There's one more question, and then we'll let you move forward. Yuzu asks if these bins will overlap, and I think the answer is yes, but go ahead.
No, the bins are not overlapping, but when the input of the network, they are overlapping. So these bins are not overlapping. So these are like, I don't know, 50 base pair non-overlapping bins. But when you introduce this flanking region for the input, then the input are intersecting. Should I continue, or there are?
Yes, please go ahead.
Okay. And I cannot, okay. So this is like the model that we use to study these RNA binding proteins, and it consists of two modules, one sequence module that extract features from the sequence information, and one region module that extract information from genomic regions, and using convolutional neural networks. And then the features from these modules are merged together and fed to another multitask module that predict the binding site of different RBPs at the same time. So, and here we define this model as a multitask approaches. So the binding site of the negative samples for one RBP is like the binding site of other RBPs. And I will show you later why this is important. So of course, at the first, we'll look into the performance of the model in the terms of the area under a precision recall curve or area under the ROC curve. AP is corresponding to area under precision recall curve. And when the scores are close to 1, it means that the performance is good. And as you can see here, the performance between different RBP varies a lot. And for example, we have some cases like muscle blind with a almost very good performance score, whereas there are some RBPs like CPS F6 that the performance is moderate. And we look into more details to see what could be the reason. And here you can see the distribution of scores for the bindings, the positive samples and negative samples of muscle blind. So the green shows like the negative samples. As you can see, the model score the negative samples close to 0, which we expect, and the positive samples close to 1, and each of them has a tail, which could represent the false positive and false negative. However, when you look at the distribution of the scores for CPS F6, which has not a very good performance, you can see that you cannot really see the clear distinction that you see here. And to investigate this in more detail, we'll look into the 6-mers for peaks that are here, like with the from the positive samples, with the lowest score and with the highest score, as well as here for the muscle blind. And we observe that the 6-mers that are enriched in the highest score binding site or highest score positive samples, you can see the right motif here. So I'm not going to do detail, but this is the case also for CPS F6, but it's not the case for the lowest scoring one. And most of them are like a UA rich element, and this is a known CLIP bias, I would say.
So there are 2 questions that pertain to the slide, so I'll go ahead and ask them. So Yaron asks if the frequency of peaks shared by more than one RBP, like at what frequency peaks are shared by multiple RBPs? And then Yosef asks if you consider cell condition, that is CLIP data is cell specific. So do you see performance of RBPs that maybe share peaks perform similarly, or what's your take on these questions?
I have like one slide in my supplementary, but we look into the intersection between the bins, and I will show you maybe later. But we don't really see many intersection between the RBPs. And the one that shared a lot of sharing binding site are mostly from the same family. So, for example, for the class of allele family, we have lots of shared binding site with the same motif, I would say. But this is not the case for like 2 different RBPs with 2 different motifs.
Okay.
And the other question, sorry, I forgot.
So CLIP data is cell specific, right? So all these results are in specific cell types, right? Or do you think they can be
Yes, actually,
generalized?
for each cell line, we train different models actually. So for each cell line, we train like 3 to 5, depending on the PARCLIP or eCLIP, because we wanted to account for the imbalanced data, because the number of the peaks for this RBP can vary a lot, from like 1,000 to millions, and it's not good for the deep learning to have like this imbalanced data. So we divided the data to 3 or 5 data and train a different model for each set.
Okay. Go ahead.
So yeah, the main concept here was that we believe that the performance score of these models are not a good proxy for a good or a bad model, and therefore, because many of this data contains lots of false positives, and these scores doesn't really like is not a good proxy for having when you have a like noisy data. Therefore, we're more interested in the interpretation of these approaches. And as I mentioned before, we use the methods that provide a model interpretability to determine, for example, in this image, we go back to the input and find the features in the input that were important for this model to classify this image as camera. And we use the same method like called integrated gradients to go back to the input sequences. So here you can see there's one input, and then find the nucleotides that were important for the model to classify this input as a binding site for example, PAM2. And as you can see, very interestingly, we can see that the right motif has been popped out. And then we can see also different kind of motifs. So we have like simple motifs, like in the case of PAM2. We also have like repeated pattern, like when you see like the same motif, very close approximity. And we also have like some cases like LF2 that bind to, like, for example, the U-rich element and with the different lengths. And we also find this complex pattern when we can find like not just one, but actually like 2 binding sites. And this CPSF6 actually is a cleavage factor, and this AAUAA is a polyadenylation signal, and this is not actually the binding motif. And so we were happy that we can locate the motif inside the input and also score like how each nucleotide is important. Here you can see we did some post-processing to find the part of this attribution maps that that has a highest score to summarize the result of the different or many attribution maps. And here you can see that the actual summary of this, or the consensus motif, is really similar to the actual known motif. And this is the case for many other RBPs as well. So we look into different RBPs, and you can see that you can find different kind of motifs, like, for example, this is like dinucleotide patterns and so on. As I mentioned before, we designed this model as a multitask approaches. So we learned the prediction by the binding site of different RBPs at the same time. And here we compared like the performance of like the multitask approach with single task approaches. And we observed that when you have like single task approaches, or you can like have different negative samples, and there are, there are different approaches to get these negative samples. You can just like randomly select from the binding site of RNA from other RBPs, or just randomly from the transcript. And if you compare the score or the performance score of these approaches, as you can see, multitask approach helps, but not that much. But if you look into the interpretation of these inputs, then you can see a clear advantage of multitask model. So here you can see, like in the two example of LF2 binding site, which binds to U-rich element. And as you can see that in the multitask model, it's really clear that the U-rich element is the motif. But when you come to the down here, this motif is almost hidden. And we can see it's like this G that is a known bias in the eCLIP data that the RNase T1, I think, clips after guanine, and this is a known bias for eCLIP par-clip data.
So I'll ask a question that is related to this point. So Irene asks, RBPs like MBNL1 and eLF2 have multiple RNA binding domains. So are you able to account for multiple RBD binding?
So you mean like different motifs, or?
So I think, yeah, go ahead.
So I didn't really get the question, because there, as long as I know, there are just like for LF2, there is like U-rich or UA-rich element, and we can find this. And also for muscle blind, there are like just one. So I don't know what was the question.
So the question is so I'm sorry, I don't know exactly. So Irene, if you can provide a bit more detail or example of the RNA binding domain. But I think from what I think this means.
You mean that it binds to different regions?
Okay, so Irene says not necessarily different motifs, but engaging longer stretches of the pre-mRNA. So MBNL1 has 4 zinc fingers. So if you see enrichment for multiple, so repeated patterns basically.
Mm-hmm. Yeah, as you can see here, we can distinguish this, yes. So.
Okay. Okay, good.
So it doesn't matter if there is a like, yeah, as you can see, we didn't specify anything. So the model has learned this motif itself. And this is like the part that was important for the prediction. So that we believe that these were like the possible binding site, but we cannot really distinguish which one.
Okay.
If the resolution of the peak or the par-clip or eCLIP is enough, then we can actually determine which one was the actual binding motif.
Okay, and so these are questions related to modeling. One of them is, why did you choose to separate the sequence and the region type modules? And then an addendum to that question, Caleb asks whether you found that specific regions were more or less informative for different RBPs.
So the first question was about the model. And so I think there is no other way to like integrate this data. So I don't know if there is a way.
So their suggestion is that you could have added the regions as additional channels to your sequence module, for example, and then trained a single model. Did you try that? Was there?
Yes, we use some structure that has like encoding for for in different areas and something like this, but it didn't work, or it didn't really help. It's
okay.
like the same.
Okay, I think those were all the questions, so go ahead.
And okay, and then so the ultimate goal of this project is to identify and score variants that with potential impact on RBP binding, and to see if the model can detect detect mutation that alter the binding site. We first use the wild type and mutated reporter construct with the known differences in binding efficiency of two RBPs. Here you can see one example from LL1 where, so to do so, we first give the model the wild type sequence and also the mutant sequence, and predict the output, and then come back to the input and visualize this attribution maps. And as you can see, by comparing the wild type with the mutant attribution maps, we can see that if in the wild type there is a long stretch of U-rich element, and when you mutate this part, this binding part is gone, and this is also in agreement with the experiments because they also detected some shift of binding to the upstream part. And in another example from Quicken 2 mutant sequences were generated targeting two potential binding of this protein. And we also did the same thing. So we give the model, the wild type and the mutant sequences, and they compare their corresponding attribution maps. And you can see here, when in the wild type we have both binding, and when you mutate one, the binding site is gone, but the other one is there. And if you mutate both binding in the mutant 2, the binding site is gone. So we took one step further and used this method as a potential tool to find and to identify and score a sequence variants that cause diseases. And to do so, we use sequence variants from cosmic data, which is a sequence variants related to the cancer. And we, for example, here we find single nucleotide variants that could destroy the binding site of muscle blind, for example, here. So here, just like C to U can destroy this binding site for this RBP. And the nice thing is that it is a robust. So when you have two different binding sites close to each other, and if you mutate one, the score doesn't change that much. It means that it can bind also to other one. And it also, there are some cases when just one single nucleotide can generate single variants, can generate a new binding site. So here in the wild type, there is no binding site, but when you mutate this C to G, it creates a new binding site. And we could find and score this sequence variants for RBPs with a good performance, because there should be the model should has learned the motif and with a good performance. So until here, I showed you the result from the ParkLib data and the model that is trained on ParkLib data. And however, when you train this models, it's a classification task. So it should be able to distinguish between binding site of one RBP, like from other cell type or from other experiments. So to assess this ability of the model, we use the data, the RBPs that are shared between the ParkLib and eCLIP, and we use the model that were trained on the ParkLib to score the peaks that obtained from the eCLIP. So the model takes as input the peak and gives some score indicating how confident the model is that the peak is a binding site of that protein. And then if we looked into the 6 mercons in the high confidence and low confidence binding site, then we observe that again, the right motifs are enriched in this high high score or high confidence binding site. And this is not the case for low confidence binding site. And this is the case for different RBPs. And this may suggest that many of the peaks that are called from PARCLIP or eCLIP could be a false positive. So here I'm showing you the attribution maps for RNA binding proteins from eCLIP data. And here you can see that, Here you can see that the ENCODE also have some in vitro experiments as well as in vivo experiments. And you can see here the motifs obtained from these two experiments, and the consensus motif that we obtained from these attribution maps is very, very similar to the ones that are reported here. And this gives us some hope that we can extend our method to a good motif finder later.
So Mahsa, there are a couple of questions about the interpretation or attribution maps. So the first question is from Yusuf Barash. So the multiple patterns, this relates to Irene's question before. So the multiple patterns you show in your examples are along the same sequence. So for example, for MBNL1, we saw the YCAY repeats. So do you identify different subtypes of motifs for the same RBP? And if you do, how do you go about defining these separate different motifs for the same RBP in your attribution maps or the PWMs that you show? And then Yusuf gives an example. A good example is the distinction recently reported by Chris Birch's group for FOX secondary motifs, or the LSAR complex, where FOX regulation can be mediated by other RBPs. So the question is, if there are different subtypes of motifs to which an RBP can bind, do you identify all of them, and how do you represent those? And I'll add on to this another question that Yusuf asked before, which is, when you said you were looking at incorporating structure to your model, would that be computationally or experimentally determined structures?
So for the first question, here is like an example. So this is not actually the binding site for this motif, but if there is also a binding site with different motif, I believe that the method can detect that. And so I want to emphasize that we don't say that this approach that we are using is like a motif finder, because we need to do more to get to that point. But here you can see that we can also find this motif, like different motifs, and also in our summary result or our consensus motif. So this is not the binding site, but we couldn't find in our data sets or a RBP that binds to different motifs. And actually, we are working on one RBP that may be like binds to different motifs in different regions, but it's not, the result is not there yet.
Okay. And what about the structures that you are planning to include?
So we are trying different kind of structures. So we are using different software to predict the structure, and also the experimental data that's already available for the structure. And so it's not just like one approach. We are trying to do different approaches and to find which one is better, but it's not really trivial, and it's really hard to predict the structure actually.
Yeah, it is. So 2 more questions pertaining your models. One is from Denise Tuma, and that is, how do you select the CNN architecture? Did you use an existing one or design your own? And how did you select the architecture hyperparameters like layers, pooling layers, which filters to use and things like that?
So yeah, in deep learning, there is no gold standard for the structure. And I think for each data, you have to find your own parameters. So you can use some cross-validation to find the best hyperparameters for your model. But as I mentioned here, I didn't really want to improve the scores, like the prediction scores, because when you have like very noisy data, this performance score doesn't give you a true proxy of how good the model is. So we like choose the hyper parameters that gives us a reasonable performance score, and then we stopped and just look into the interpretation.
Okay, good. So Yaron asks, how well does your model trained on PowerClip data generalize on the eCLIP? And I want to add to that question. So when you're looking at CLIP data, PowerClip data and eCLIP data, are these across the same cell line?
Here, when I showed you this slide, so no, these are not from the same cell line. So all PowerClip data are from hex cell line, and the eCLIP data are from K562 and HepG2. So they are not from the same cell line, and I don't have the performance here, because again, I don't really believe in the performance score when you have very noisy data, and we know that eCLIP also is very noisy. So here we, to assess this, we just look into the 6 merge cons from high scoring and low scoring binding sites. And we observe that actually the high scoring binding site actually contains the right motifs or enriched for the right motifs. And this gives us some confidence that the model has learned the true features, and it can also generalize to other data.
So, but do you think, so for example, if there are, do you see any correspondence between overlap of targets of that an RBP binds to across cell lines and how well your model performs for those RBPs compared to RBPs that do not share peaks across cell lines? And if you can tell us if that affects the generalization part.
So I didn't really look into how the intersection between cell lines works. So I don't know how the PowerClip and eCLIP, for example, this case are intersecting, because I think the expression, like the cell line have different gene expression, so it's not really comparable, because the RNA is different, the transcript is different. But when you when you train a model or a classification model, then, and if you are sure that the model has learned the true features, it shouldn't be a problem for generalize it to other cell lines or other
but you would think that the context matters, right? If you generalize it and try to generalize it to an extremely dissimilar cell lines, I'd be very surprised if the model performs really well, because the context has changed.
So I would say that it depends on the RBP. If there is a clear motif, like, I don't know, PAM2, and if you know the transcript in your cell line, then it's fine. But for example, there are some RBPs that the known, like the motifs is not really known, or there is no sequence motif, then yeah, I wouldn't say that the model can find anything. Of course, there are some differences between cell lines, and maybe it cannot find all possible binding sites, but yeah, I think it's not a problem when there is a clear motif, for example, in the case of PAM2.
Okay, I think that's all for now.
So yeah, then it comes to the summary, and so we develop a multimodal and multitask deep learning approach to model the genuine and specific RBP binding events. And we could also use this noisy clip data sets, and we also could extract some informative features about this RNA binding proteins characteristic using the interpretation of these models. And we hope that we could extend these methods to a motif finder, but now also the methods provide some functionality to locate binding site and drive motifs from attribution maps. And the interesting is that it can be used potentially to identify and score non-coding sequence variants that alter RBP binding site, and we are working on this and to improve the methods and account for many problems that may occur to reach this goal. And with this, I would like to acknowledge my supervisor, Uwe Ola, and Neil, a former postdoc in our group that provided the ParkLab data, and of course, all people in the Ola lab for a nice atmosphere and discussion and everything. And with this, I would like to thank you for your time and listening to my talk, and I'm happy to get more questions.
Okay, let's everybody thank Mahsa, and I'll clap on everyone's behalf.
Thank you.
So there's one more question for you, and I'm really happy that we have had so many good questions. So we have some time, so if others have more questions, feel free to type them in the Q&A section. So this question is, to define the regions for the 50 base pair bins, do you consider alternative isoform annotations, or do you collapse the transcripts on the longest isoform?
So we didn't do any isoform detections. So this is what I'm working now. So to find the isoform with highest expression and just use this annotation. But for these models, we just use the annotation that so that there are some overlap, and we just use one. So regardless of the expression values or isoforms.
Okay. Okay. That's interesting. Okay, there's one more question. Okay, so in this work, you have used integrated gradients for interpretation. So there is, Yusuf Barash asks that there was recently a discussion about how well different interpretation methods perform on different data types. So do you have any observations regarding that? Did you try others?
Yes, actually we tried this when we were working on these methods. And at the time that I was working on this, there were some other methods like DeepRiPe, and we compare the other different methods. And for our task, the results were so similar, so that we decided to use this integrated gradients because it doesn't depend on the structure, and it can be used for also recurrent neural networks. Yeah, we compared this. I forgot to. I wanted to had the slides there, but we compared them, and it was really, really similar. So I think in the case of for example, this RBP, and because the model is not that deep, and also the input is not that long, so different methods we also use other methods like LRP, or that that is also people here in Berlin have introduced that, and all of them have very similar results. And we are working, and hopefully with others, to find the best attribution methods for genomics applications, and also some to have some benchmark to assess this, because it's not really straightforward to assess this systematically which one is the best one.
Okay, so there's one more question from Denise. Did you experiment with different bin sizes? And I think that's straightforward to answer. Did you experiment with different bin sizes?
Yes, so we defined the bin sizes based on the length distribution of the peaks in each data sets. So for Parklib, the bins are not that long, so it's like around 50 base pair, and that was the reason that we chose 50 for eCLIP, Parklib data. But for eCLIP data, the lengths of the peaks are bigger, so it's like around 200, I would say, or 100. So we chose like 100 length bin. So I think it depends on the data, and yeah, you can change this. But
okay, good. Yeah, I think that's all the questions that we have had. So thank you once again, Mahsa, for this wonderful talk.